Hello and welcome, dear students. Today we are going to discuss an important topic, that is historical perspective of cell theory and exceptions to the cell theory. The main objectives of today's lecture are to elaborate historical backgrounds in the discovery of cells, to elaborate the historical backgrounds in the discovery of various cell organelles, to discuss the cell theory in detail, to explain some of the fundamental properties of the cell, to explain the various exceptions of the cell theory. Dear students, let me give the introduction. Cells and the structures they comprise are too small to be directly seen, heard, or touched. In spite of this tremendous handicap, cells are the subject of hundreds and thousands of publications each year, with virtually every aspect of their minuscule structure coming under scrutiny. This is not to imply that cells and molecular biologists have a monopoly on these noble traits. Cell and molecular biology is reductionist, which means it is based on the view that knowledge of the parts of the whole can explain the character of the whole. When viewed in this way, our feeling for the wonder and mystery of life may be replaced by the need to explain everything in terms of the working of the machinery of the living system. Now let us discuss about the historical perspective of cell. Because of their small size, cell can only be observed with the aid of microscope. We don't know when humans discovered the remarkable ability of curved glass surfaces to bend light and form images. Spectacles were first made in Europe in the 13th century, and the first compound light microscope were constructed by the end of the 16th century. By the mid 1600s, a handful of pioneering scientists had used their handmade microscopes to uncover a world that would never have been revealed to the naked eye. The discovery of cell is generally credited to Robert Hooke, an English microscopist who at the age of 27 was awarded the position of curator of the Royal Society of London. In actual fact, Hooke had observed the empty cell walls of dead plant tissue, walls that had originally been produced by the living cells they surrounded. Meanwhile, Anton van Leeuwenhoek, a Dutchman, was spending his spare time grinding lenses and constructing simple microscopes of remarkable quality. Leeuwenhoek was the first to examine a drop of pond water under the microscope, and to his amazement, he observed the teeming microscopic animalcules. He was also the first to describe various forms of bacteria. His initial letters to the Royal Society describing this previously unseen world were met with such skepticism that the Society dispatched its curator, Robert Hooke, to confirm the observations. Now we will talk about the discovery of various cell organelles. Cell membrane. Plasma membrane encloses cell and represents its outer living boundary. This membrane was called cell membrane by C. Nageli and C. Kramer in 1855 and plasmalemma by J. Q. Plover in 1931. On the exterior of this membrane is present extracellular matrix, which is an organized network of extracellular materials. It often plays a key regulatory role in determining the shape and activities of cell. For example, enzymatic digestion of ECM that surrounds cultured cartilage cell and mammary gland epithelial cells cause a marked decrease in the synthetic and secretory activities of the cell. Mitochondria. The first observation of intracellular structures that probably represented mitochondria were published in 1840s. Richard Altman in 1890 established them as cell organelles and called them bioblasts. The term mitochondria was coined by Carl Benda in 1898. In 1904, Mavis made the first recorded observation of mitochondria in plants in cells of the white water lily Nymphia alba. Benjamin F. Kingsbury in 1912 first related them with cell respiration, but almost exclusively based on morphological observations. In 1930, particles from extracts of guinea pig liver were linked to respiration by Otto Heinrich Warburg, which he called grana. Warburg and Heinrich Otto Wieland, who had also postulated a similar particle mechanism, disagreed on the chemical nature of the respiration. It was not until 1925 when David Keeling discovered cytochromes that the respiratory chain was described. Plastids. The term plastids is derived from Greek word plastikos meaning formed or molded, and was used in 1885 by A. F. W. Skimper to include cell organelles which primarily involved in formation and storage of soluble and insoluble carbohydrates. In favor of term plastids, the term chromatophore, earlier used for green pigments in leaves, was abandoned. This was necessary because it was discovered that these organelles could also be colorless, orange or yellow, besides their common green color. These are autonomous structures and would never arise de novo. Depending upon the presence or absence of pigments, Skimper classified the plastids into colorless leucoplasts, colored chromoplasts, and green chloroplasts. Skimper also suggested different kinds of plastids can transform into one another. Plastid inheritance independent of nucleus is also present in the cell. Plastid carrying the genetic information is collectively called plastidome. Daengard, 1920. Endoplasmic reticulum. The endoplasmic reticulum were first seen in 1945 by Keith R. Porter, Albert Claude, Brodie Meskers, and Ernest F. Fulham using electron microscopy. The endoplasmic reticulum is a type of organelle in eukaryotic cells that forms an interconnected network of flattened membrane enclosed in sac or tube-like structures known as cisternae. The membranes of endoplasmic reticulum are continuous with the outer nuclear membrane. The endoplasmic reticulum occurs in most cell types of eukaryotic cells but is absent from red blood cells and spermatozoa. There are two types of endoplasmic reticulum, rough and smooth. The outer, that is cytosolic face of the rough endoplasmic reticulum is studded with ribosomes that are the sites of protein synthesis. The smooth endoplasmic reticulum lacks ribosomes and functions in lipid manufacture and metabolism, the production of steroid hormones and detoxification. Ribosomes. The ribosomes were first observed in mid-1950s by Romanian cell biologist George Emil Palade using electron microscope as dense particles or granules. The term ribosome was proposed by scientist Richard B. Roberts in the end of 1950s. A ribosome is made from complex of RNAs and proteins and is therefore a ribonucleoprotein. Each ribosome is divided into two subunits. A smaller subunit which binds to larger subunit and the mRNA pattern, and a larger subunit which binds to tRNA, the amino acids, and the smaller subunit. Ribosomes are ribozymes because the catalytic peptidyl transferase activity that links amino acids together is performed by the ribosomal RNA. Golgi complex. Owing to its large size and distinctive structure, the Golgi apparatus was one of the first organelles to be discovered and observed in detail. It was discovered in 1898 by Italian physician Camillo Golgi during an investigation of the nervous system. Some doubted the discovery at first, arguing that appearance of the structure was merely an optical illusion created by the observation techniques used by Golgi. With the development of modern microscopes in the 20th century, the discovery was confirmed. The term Golgi apparatus was used in 1910 and first appeared in the scientific literature in 1913. The Golgi apparatus packages proteins into membrane-bound vesicles inside the cell before the vesicles are sent to their destination. It is of particular importance in processing proteins for secretion, containing a set of glycosylation enzymes that attach various sugar monomers to protein as the proteins move through the apparatus. Lysosome. Lysosome is membrane-bound organelle found in nearly all animal cells. They are spherical vesicles which contain hydrolytic enzymes that can break down virtually all kinds of biomolecules. The lumen's pH 4.5 to 5.0 is optimal for the enzymes involved in hydrolysis, analogous to the activity of the stomach. Besides degradation of polymers, the lysosome is involved in various cell processes including secretion, plasma membrane repair, cell signaling, and energy metabolism. By 1949, Christian Didier and his team had focused on the enzyme called glucose 6-phosphatase, which is the first crucial enzyme in sugar metabolism and the target of insulin. They already suspected that this enzyme played a key role in regulating blood sugar levels. However, even after a series of experiments, they failed to purify and isolate the enzyme from the cellular extracts. Therefore, they tried a more arduous procedure of cell fractionation by which cellular components are separated based on their sizes using centrifugation. They succeeded in detecting the enzyme activity from the microsomal fraction. This was the crucial step in the serendipitous discovery of lysosomes. To estimate this enzyme activity, they used that of standardized enzyme acid phosphatase and found that the activity was only 10% of the expected value. One day, the enzyme activity of purified cell fractions, which had been refrigerated for 5 days, was measured. Surprisingly, the enzyme activity was increased to normal of that of the fresh sample. The result was the same no matter how many times they repeated the estimation and led to the conclusion that membrane-like barrier limited the accessibility of the enzyme to its substrate, and that the enzyme were able to diffuse after a few days and react with their substrate. They described this membrane-like barrier as a sac-like structure surrounded by a membrane and containing acid phosphatase. Peroxisomes. Peroxisomes were identified as organelles by Belgian cytologist Christian Didier in 1967 after they had been first described by a Swedish doctoral student J. Rodin in 1954. They are involved in catabolism of very long-chain fatty acids, branch-chain fatty acids, D-amino acids, and polyamines, reduction of reactive oxygen species, specifically hydrogen peroxide, and biosynthesis of plasmalogens. Other functions of peroxisomes are photorespiration in leaves, glycolysis in trypanosomes and methanol, and amine oxidation and assimilation in some yeasts. Centrosomes. It was discovered by Edward van Beneden in 1883 and later described and named in 1888 by Theodore Boveri. In cell biology, the centrosome is an organelle that serves as the main microtubule organizing center of the animal cell as well as a regulator of cell cycle progression. Fungi and plants lack centrosome and therefore use other MTOC structures to organize their microtubules. Although the centrosome has a key role in efficient mitosis in animal cell, it is not essential in certain fly and flatworm species. In general, each centriole of the centrosome is based on 9 triplet microtubule assembled in a cartwheel structure and contains centrin, connexin, and tectin. In many cell types, the centrosome is replaced by cilium during cellular differentiation. Nucleus. The nucleus was first organelle to be discovered. What is most likely the oldest preserved drawing dates back to early microscopist Antony van Leeuwenhoek, 1632 to 1723. He observed a lumen, the nucleus in the red blood cells of salmon. The nucleus was also described by Franz Baer in 1804 and in more detail in 1831 by Scottish botanist Robert Brown in a talk at the Linnean Society of London. In 1838, Matthias Seclidin proposed that the nucleus plays a key role in generating cells. Thus, he introduced the name cytoblast, that cell builder. The idea that cells can be generated de novo by cytoblast or otherwise contradicted work by Robert Remarque, 1852, and Rudolf Virchow, 1855, who decisively propagated the new paradigm that cells are generated solely by cells, omnis cellulae cellula. Between 1877 and 1878, Oscar Hertwig published several studies on fertilization of sea urchin eggs, showing that the nucleus of the sperm enters the oocyte and fuses with its nucleus. In 1873, August Weismann proposed the equivalence of maternal and paternal germline cells for heredity. After mitosis was discovered and the Mendelian rules were rediscovered at the beginning of the 20th century, the chromosome theory of heredity was therefore developed. Let us now discuss about the cell theory. Credit for developing cell theory is usually given to two scientists, Theodor Schwann and Matthias Jakob Seclidin. While Rudolf Virchow contributed to the theory, he is not as credited for his attribution towards it. In 1839, Seclidin suggested that every structural part of a plant was made up of cells or the result of cells. In 1839, Theodor Schwann stated that along with plants, animals are composed of cells or the product of cells in their structures. This was a major advancement in the field of biology since little was known about animal structure up to this point compared to plants. From these conclusions about plants and animals, two of the three tenets of the cell theory were postulated. All living organisms are composed of one or more cells. The cell is the most basic unit of life. In 1855, Rudolf Virchow added the third tenet to cell theory. In Latin, this tenet states omnis cellulae cellula. This is translated to all cells arise only from pre-existing cells. However, the idea that all cells came from pre-existing cells had in fact already been proposed by Robert Remarque. Remarque published observations in 1852 on cell division, claiming Seclidin and Schwann were incorrect about generation schemes. He instead said that binary fission was how reproduction of animal cells were made. Once this tenet was added, the classical cell theory was complete. We shall now discuss about the modern interpretation of cell theory. The generally accepted parts of modern cell theory include all known living things are made up of one or more cells. All living cells arise from pre-existing cells by division. The cell is the fundamental unit of structure and function in all living organisms. The activity of an organism depends on the total activity of independent cells. Energy flow, metabolism and biochemistry occurs within cells. Cells contain DNA, which is found specifically in the chromosomes, and RNA found in the cell nucleus and cytoplasm. All cells are basically the same chemical composition in organisms of similar species. Students, we will now discuss about the modern version of the cell theory, which includes the idea that energy flow occurs within the cells. Heredity information DNA is passed on from cell to cell. All cells have the same basic chemical composition. We will discuss about some of the basic properties of cells. Just as plants and animals are alive, so are the cells. Life, in fact, is the most basic property of cells, and cells are smallest unit to exhibit this property. Remarkably, cells within the body generally die by their own hand, the victims of an internal program that causes cells that are no longer needed or cells that pose a risk of becoming cancerous to eliminate themselves. The first culture of human cells was begun by George and Martha Gay of Johns Hopkins University in 1951. The cells were obtained from a malignant tumor and named HeLa cells after the donor Henrietta Lacks. HeLa cells descended by cell division from this first cell sample are still being grown in laboratories around the world today. The following are some of the basic properties of cells. Cells are highly complex and organized. Complexity is the property that is evident when encountered, but difficult to describe. Cellular activities can be remarkably precise. DNA duplication, for example, occurs with an error rate of less than 1 mistake every 10 million nucleotides incorporated, and most of these are quickly corrected by an elaborate repair mechanism that recognizes the defect. There is a great deal of consistency at every level. Each type of cell has a consistent appearance when viewed under high-powered electron microscope. Similarly, each type of organelle has a consistent composition of macromolecules which are arranged in a predictable pattern. For example, each mitochondrion is composed of a defined pattern of internal membranes, which in turn are composed of a consistent array of proteins, including an electrically powered ATP synthesizing machine that projects from the inner membrane like a ball on a stick. Cells possess a genetic program and the means to use it. Organisms are built according to information encoded in a collection of genes. Remarkably, this vast amount of information is packaged into a set of chromosomes that occupies the space of a cell nucleus. Genes constitute the blueprints for constructing cellular components, the directions for running cellular activities, and the program for making more of themselves. Cells are capable of producing more of themselves. Just as individual organisms are generated by reproduction, so too are individual cells. Cells reproduce by division, a process in which the contents of a mother cell are distributed into two daughter cells. Prior to division, the genetic material is faithfully duplicated and each daughter cell receives a complete and equal share of genetic information. In most cases, the two daughter cells have approximately equal volume. In some cases, however, as occurs when a human oocyte undergoes division, one of the cells can retain nearly all of the cytoplasm even though it receives only half of the genetic material. Cells acquire and utilize energy. Every biological process requires the input of energy. Virtually all of the energy utilized by life on the Earth's surface arrives from the electromagnetic radiations from the sun. The energy of light is trapped by light-absorbing pigments present in the membrane of photosynthetic cells. Light energy is converted by photosynthesis into chemical energy that is stored in energy-rich carbohydrates such as sucrose or starch. For most animal cells, energy arrives prepackaged, often in the form of sugar, glucose. Cells expend an enormous amount of energy simply breaking down and rebuilding the macromolecules and organelles of which they are made. Cells carry out a variety of chemical reactions. Cells function like miniaturized chemical plants. Even the simplest bacterial cell is capable of hundreds of different chemical transformations, none of which occur at any significant rate in the inanimate world. Virtually all chemical changes that take place in cells require enzymes, molecules that greatly increase the rate at which a chemical reaction occurs. The sum total of chemical reactions in a cell represents that cell's metabolism. Cells engaged in mechanical activities. Cells are site of bustling activity. Materials are transported from place to place, structures are assembled and then rapidly disassembled, and in many cases the entire cells move itself from one site to another. Cells are able to respond to stimuli. Some cells respond to stimuli in obvious ways. A single-celled protist, for example, moves away from an object in its path or moves towards a source of nutrients. Cells within a multicellular plant or animal respond to stimuli less obviously. Most cells are covered with receptors that interact with substances in the environment in highly specific ways. Cells may respond to specific stimuli by altering their metabolic activities, moving from one place to another, or even committing suicide. Cells are capable of self-regulation. In addition to requiring energy, maintaining a complex ordered state requires constant regulation. The importance of cells' regulatory mechanisms become more evident when they break down. We are gradually learning how a cell controls its activities, but much more is left to discover. Evolution of the cells. It is presumed that cells evolved from some type of precellular life form, which in turn evolved from non-living organic materials that were present in the primordial seas. Whereas the origin of cells is shrouded in near total mystery, the evolution of cells can be studied by examining organisms that are alive today. There are some of the exceptions to the cell theory. Life does not have a hard and fast definition, so it is impossible to identify something that everyone will recognize as both living and non-cellular. However, there are few exceptions to the cell theory as follows. All living organisms are made up of one or more cells. An exception to this would be known as non-cellular life. The strongest candidates for non-cellular life includes viruses, viroids, and prions. Viruses are non-cellular entities most often considered potentially alive. They carry genetic material, they evolve, and they use various proteins as their machinery of action. However, they are unable to metabolize and produce their own proteins. They depend on infected cells doing all the work for them. Because they are incapable of acting on their own, they are not considered living by many. The counter-argument is that all life ultimately depends on external factors for survival, so viruses depending on cells for living may not be a disqualifier since they still control the processes resulting in their reproduction. Unlike viruses, viroids lack any proteins whatsoever. As a result, even fewer people are willing to consider viroids alive. However, the same logic used to argue the biotic nature for viruses can be used for viroids. They copy themselves the same way many RNA viruses do. If a viroid obviously is not life, viruses must be held suspect since they can be argued similar to viroids than cellular life. Prions are interesting. They do not reproduce like viruses or cell. That is to say, they do not direct cellular machinery to construct copies of themselves. Instead, they modify existing proteins they come in contact with. Back and forth arguments aside, it has become fairly common to view viruses as a sort of middle ground between life and non-living processes, not one or the other. Cells are the smallest unit of life. This is not quite separate from number 1 and 3. Any non-cellular life form could be used to argue that the cell is not the basic unit of life. Similarly, a cell not originating from a pre-existing cell could be used to make a similar argument, because it is highly unlikely that all the processes found even the most basic cell could start from scratch. Of course, we can also use the above points to argue that the cell is indeed the smallest unit of life. Viruses, viroids, and prions can be used to support the assertion that cells are the smallest unit of life. Even though they are not cells, their reproduction is only made possible by the activities carried out within the cells. Things like RNA world only would have required the processes for life gradually building up from less ordered, spontaneous reactions. An RNA world could be argued as a world devoid of true life, since life can be argued as things which carry out several key processes and not as merely the individual processes themselves. Cells arise from pre-existing cells. This one is the only easy one. This had to be violated by abiogenesis. There had to be the first cell. The origins of the first cells are variously explained by RNA world, GADV protein world, and other hypotheses. This means there must be one or more cells in the history of life that were spawned from some natural processes lacking a parent cell. We can view the cells from Cell's rule a bit like Newtonian gravity. It is mostly right, with its fault only becoming obvious under conditions not readily present on Earth. With this, we end our today's program. Thanks for watching.